Have you ever just stared at a plant, maybe a really rare one, and just wondered, what kind of secrets are locked away inside its cells? Well, what if I told you that from just one tiny, almost invisible piece of that plant, you could grow an entire, perfect copy? Today, we're going to do just that. We're unlocking the mystery.
You see, the real magic, the incredible potential for life, it all happens on a scale that most of us just never get to see. It's hidden away in every leaf, every stem, every seed. It's a microscopic world of possibility, just waiting for the right moment to burst into life.
So, let's just ask the big question. What if you could become a kind of guardian for nature's coolest creations? A cloner of rare beauty, all from your own home? I know, it sounds like science fiction, right? But it's not. It's the very real and totally fascinating world of plant tissue culture. And our journey into it, well, it starts now.
To understand this, we have to start small. It all begins with the very foundation of life itself, the single cell.
So what are we really talking about here? Plant tissue culture is basically the art and science of growing plants in glass. The whole thing hinges on two absolutely critical things. You need perfectly sterile conditions, and you need to provide a custom-made nutrient medium. If you can nail those two, you can unlock a plant's secret ability to regenerate from the tiniest little piece.
And when we say sterile, we really mean it. This is the first and, honestly, probably the biggest challenge. Any piece of a plant that you use, we call it an explant, it has to survive this intense cleaning process. It's a strict sequence. First, an alcohol wash, then a good soak in a bleach solution, and finally, several rinses in sterile water to wash all that off. There are no shortcuts here. This step is completely non-negotiable.
Okay, so once you have a squeaky clean explant, it needs a home. So, welcome to the lab kitchen, where we start mixing up what are basically elixirs of life.
And this takes some serious precision. This here is a classic recipe. It's called Murashige Skoog, or just MS Media for short. It's this specific cocktail of mineral salts for nutrition, sugar for energy, and then agar to get that jello-like texture. And check this out. Even the pH has to be dialed in perfectly to 5.7. It's like you're cooking a gourmet meal for a guest that's, microscopic.
But this is where things get really cool. This is where we go from being a cook to being a kind of chemist. By adding specific plant hormones, we can actually tell the plant what to do. You've basically got two main options. To create this blob of undifferentiated cells we call a callus, you use a lot of a hormone called auxin. But if you want that callus to actually sprout new shoots, you flip the script. You use way more of another hormone, cytokinin. It's literally like giving the cells a set of instructions.
So when you look at that finished gel, what is it actually made of? Well, it's mostly water. Then you've got your essential salts and sugars. But look closer. That gelling agent and those super powerful hormones we just talked about, they make up just a tiny, tiny fraction of the whole thing. But believe me, that small percentage, it makes all the difference in the world.
Now, everything we've talked about so far sounds pretty straightforward, right? Mix this, add that. But every single person who does this knows there is a constant, invisible war being fought inside every single one of these jars. This is the battle for purity.
The experiment logs we're pulling from for this explainer are full of amazing successes, but they're also brutally honest about the failures. This entry right here, translated from the original, just says it all. Result, everything died. It didn't work this time. It's a humbling and honestly a pretty common experience in tissue culture.
And the enemies are literally everywhere. You've got the classic green mold, bacteria, and yeasts just floating around in the air, waiting for a chance to land on that rich, sugary food source. But then you have the sneakier stuff, like contamination that's already hiding inside the plant tissue. And of course, there's always the risk of a mistake during a transfer.
And here's another entry from that log that just drives the point home. Powerful green mold contamination. It's a relentless fight. A single, invisible spore can wipe out weeks of careful, painstaking work. Every successful culture you get is a hard-won victory. But for all that struggle, those moments of success, oh, man, they make it all worthwhile. Because after the battle, you get the breakthrough, and all that hard work finally grows into something truly amazing.
After pages and pages of documenting contaminations, you find a note like this one in the log. Just 2 simple words, but they mean everything. Callus and root. That's it. That means not only did the little guy survive, but it actually followed the hormonal instructions and started building a whole new plant. It's a huge breakthrough.
And from that little moment, a new life just unfolds. This timeline, taken right from the logs, shows the journey of one single tomato seed. It took about 2 weeks just to see it sprout, another month to become a healthy little plantlet, and then, after 3 whole months in the jar, it was finally ready to move into soil. It's a process that definitely requires patience, but wow, the payoff is just incredible.
And what's really mind-blowing is just how many different things you can do this with. The person behind these logs didn't just stick to one type of plant. No way. They worked on everything from rare cacti and trendy houseplants like monsteras to flowers, vegetables, and even fungi and trees. The basic principles work across a huge part of the living world.
So we've seen the science, we've seen the struggle, and we've seen the success. But this journey isn't just about watching from the sidelines. It's about jumping in.
And just cloning a plant is really only the beginning. These techniques can sometimes cause spontaneous, natural changes. Scientists call it somaclonal variation. This opens up a whole new world of discovering or even creating brand new plant traits, making new varieties that have never existed before right there in a petri dish. This journey of discovery, it never really stops. I absolutely love this note from the experimenter's to-do list. Video how to monstera, protocols, conditions, timings. It just shows that drive to not only figure things out, but to get it just right and then share that knowledge with everyone else.
So, we've just walked through one person's incredibly detailed, passionate, and very real journey into tissue culture, all from their personal notes. It's a story of patience, of precision, and of profound discovery. And it really just leaves us with one final thought. These lab notes tell an amazing story. So, what kind of story will you create?